Alright, this is my fourth try. The first one had no audio. The second and third ones, it just didn't want to start recording. So now let's just see what we got. This chapter is going to cover Mendel's experiments in heredity, and it will also cover some non-Mendelian genetics. Mendel's experiments are like the Week 9 lab that didn't get posted till too late and is now extra credit. But it will also cover the human heredity, that is, this week's lab. And some of those types of inheritance don't exactly follow the patterns of Mendel's inheritance. So we'll go through the Mendelian inheritance first, a bit of a review for those of you who dug into lab archives and found the Week 9 lab, and for those of you who have not done so, it's not too late. And this will provide an excellent foundation for this week's lab on human heredity. So there were a lot of misconceptions about heredity before we knew about chromosomes, ranging from thinking that the sperm actually had fully formed little people inside them, that offspring were really exact duplicates from their parents, that the egg had all the traits, that the sperm had all the traits, that inheritance was blended, meaning that you're always an exact average between the parents, and that we could acquire characteristics during our lifetime, and that those acquired characteristics would be passed on to our offspring. One of the classic examples of that people used to support this concept was giraffes having long necks. The hypothesis being that those giraffes that stretch their necks to reach the taller branches and get more food made their necks longer during their lifetimes, and then their offspring would be born with long necks. Well, the thing that actually happens is that the giraffes that had long necks already were more successful. That means they had genes for the long necks. And then pangenesis, that little particles that they called gemmules carry the traits we inherit. We now know, of course, that those packets, those particles, are our chromosomes. This is an image of the homunculus, where the sperm has a fully formed little individual that's microscopic, and then it would just get bigger and become the infant that was born. This slide lists a lot of individuals that helped us come to our modern understanding of genetics. You will not be held responsible for knowing which person discovered which part of genetics. Mendel was the father of genetics because his experiments did actually show us how we can expect inheritance to work, even without knowing what chromosomes are. Mendel was a great geneticist, even though he didn't know it was genetics, but he was also really lucky. He was looking at what we call dichotomous traits, an either-or. The flowers in peas were either purple or white. There weren't any other colors. The seeds were yellow or green. The seeds were wrinkled or round. The pods were inflated or constricted. There was never any intermediate between any of those. That's what's meant by dichotomous. The true breeding traits are those that there's only one type of information to pass, and that the flowers were monoecious, meaning in a given flower there were both male and female reproductive parts, and that means that he could control by removing the male reproductive parts which pollen actually fertilized which female parts of the flower. And that each pea in a pod is an individual. So you can get thousands of individuals counted very, very easily. And the plants have very short generation times, and they're cheap and easy to grow. In our genetics notation system, we use P to denote the parent generation, the initial round. And for genetics problems, we typically, at least while discovering how the genetics work, start with true breeding plants. That means that they carry only one characteristic. If it's a purple flower, it only has genetic information for purple flowers. And if it's a white flower, it only has genetic information for white flowers. The F1 is the generation that comes from those initial parents. They're the brothers and sisters. The F2 is the second filial generation, which means if you cross two of those F1s. And with plants, you would be crossing the brothers and sisters. With animals, it's not such a great idea. And then each successive generation has the F, which stands for filial, the brothers and sisters, and which generation it is. A cross-sib is a mating between brothers and sisters. Again, not a big deal with plants that already cross-fertilize within the same plant. Not a great idea with animals. A back cross is mating an offspring back to its parent or other ancestor. Easily done with plants again. Self-crossing really only happens with plants because the flower might contain both the male and female reproductive parts, or on the same plant there are both types of flowers, female flowers and male flowers on the same plant. And those true breeding means they have the exact same traits. They don't have any combinations of traits. They don't have a purple piece of information and a white piece of information. The plant only has purple or it only has white. This is our example of looking at the true breeding parent. If they are both true breeding purple, then they only have purple genetic information, meaning their offspring can only be purple, and if we cross them, they're all going to be genetically identical. But if we cross true breeding purple with true breeding white, then we see a different result. The F1 is purple, and it looks exactly as purple as the purple parent, but they are hybrids because we mixed a true purple with a true white. And this is where the terms dominant and recessive come into play. Purple is a dominant trait and white is a recessive trait. The white information, the genetic material making that plant white, is still present. And it doesn't blend because we don't get a lavender flower when we mix purple and white. But it recedes into the background. It is recessive. And if you cross two of these plants that have a mixture of one purple piece of information and one white piece of information, and you cross them together, the F2, the second generation, looks like three perfect purples, but you get a white back out of that. And this is the type of phenomenon you may have heard people talk about when they say a characteristic or a trait in their family skips a generation. It's because recessive traits require both parents to contribute genetic information for that recessive trait in order for us to be able to see that recessive trait in the offspring. Let's see how that works out. Here is our actual cartoon looking at the same true breeding purple with true breeding white. The F1 is a mixture. If we cross two of the same F1s, the appearance will be 3/4 of them purple and 1/4 of them white, but their genetics of all the purples will not be identical. There's our dominant and recessive. The recessive trait is maintained in that F1 generation. We just don't see it. And those ratios of 3 dominant to 1 recessive said that those F1s, that first generation where they all looked purple, had to carry some hereditary particle for purple and a hereditary particle for white. And that ratio suggested that they segregated into gametes and then came back in fertilization. If you go back into week 9's genetics, Mendelian genetics lab, there is an exercise of flipping coins to represent the chances of it being a dominant or a recessive allele. And the spreadsheet that is linked in the lab archives notebook automatically tallies the sum of all the classes flips and calculates the ratios of how many would be 2 dominants, how many would be 2 recessives, and how many of those flips would be 1 dominant and 1 recessive. And Mendel did not have any idea what these particles were, but he could follow the effect of those particles by looking at the results of the offspring in the mated pea plants that he had. And those particles, of course, turned out to be chromosomes. Our vocabulary that is useful for both the Mendelian genetics and the human genetics is a trait that is a characteristic. It can be talking about non-animals like flowers. It could be talking about animals like eye color. It could be, we're going to look at human traits like tongue rolling. Being able to do that is genetic. Whether your little fingers are bent or straight. If you have a widow's peak or not. And it may not be something that you could actually physically see, but any sort of trait, biochemical trait, would also be a trait or characteristic that is genetically encoded. All of our genetic problems are going to use things we can physically see because it makes it a lot more concrete. Other terms we need for describing the process of our genetics problems is phenotype. It begins with a PH, and it describes the physical appearance, the characteristic that we actually see. So the trait is flower color. The phenotype would be purple flower, white flower. Eye color is the trait. The phenotype would be blue eyes, gray eyes, hazel eyes. And so the alleles are the specific versions of that hereditary particle. And that hereditary particle is the gene. So the gene is flower color. The allele is purple flower. The gene is eye color. The allele is green eyes. Dominant recessive refer to the alleles, the specific variations that are present in an individual. So a dominant allele will mask or overtake, overshadow the other alleles that are present. And it's typically designated with an uppercase or capital letter. So an uppercase P for purple. The recessive allele is present and useful and can be passed to offspring, but it gets masked by dominant alleles and is typically designated with the same letter as the dominant allele, but in lowercase. So not a W for white flower, but a lowercase p for the white flower. The genotype tells us the specific combination of those hereditary particles, what alleles are present. And the way we can describe our genotype is by the term homozygote. An individual is a homozygote if, for a particular characteristic or trait, the alleles they carry are exactly the same. So you can be either homozygous, that's the adjective form, homozygous dominant, having 2 dominant alleles for 1 trait, or you could be homozygous recessive, having 2 recessive alleles for 1 trait. The other type of genotype is a heterozygote. And hetero means a mix. So if you're a heterozygote, or you are heterozygous, you have 2 different alleles for 1 trait. So having a purple allele and a white allele. And so heterozygotes are always not true breeding. To be true breeding means you have to have identical alleles for a given trait. And the last thing that got slightly cut off is zygote, Z-Y-G-O-T-E. That is the first cell that is produced when the gametes fuse. After mitosis starts, you're no longer a zygote. So that's why the terms are heterozygote and homozygote. When the zygote is formed, when the gametes fuse, do we have identical alleles? Then it is homozygous. And if we have different alleles, it is heterozygous. The gene is actually a sequence of DNA. And that encodes that information that we need for the characteristic. And so the allele is the specific variation of that gene. A locus is a place on a chromosome, an actual physical location, and it is typically referring to a gene. When we look at our genetic problems, we're going to see terms like reciprocal cross. It's a form of mating cross where a previous cross is repeated, but we swap which parent has what trait. So that's saying if we had a blue-eyed father and a brown-eyed mother in the first cross, the reciprocal would be a blue-eyed mother and a brown-eyed father. And we do that kind of cross to see, does it matter if it's the male parent or if it's the female parent that is donating that information to the offspring? In most cases, it doesn't matter, but we will see types of inheritance where it will matter if it's the male or the female parent that is donating that genetic information. And lastly, on this slide, we see wild type and mutant alleles. Wild type just means it is the normal allele in the population, whereas mutant means it formed by mutation. It is not as common. So here is showing the alleles for how we got those results of the purple F1 generation and the 3-to-1 purple to white generation with the pea plants. True breeding means homozygous. True breeding purple, we are homozygous and dominant. True breeding white, homozygous and recessive. And again, we pick one letter for the characteristic, uppercase for the dominant allele, lowercase for the recessive allele, but the same letter. For a cross with just one trait, it's not that big of a deal, but as we move into more complicated genetics problems, it makes it much harder if you start using multiple letters. I'm going to stick with one letter. So if you have a true breeding dominant and a true breeding recessive, the only possible result is that a big P from this individual fertilizes a little p from that individual. We can take this big P with that little p, or the same big P with the second little p, but no matter which big P fertilizes which little p, the offspring can only have the genotype of one dominant and one recessive allele. Anytime a true breeding dominant is mated with a true breeding recessive, the only possibility for its offspring is to be heterozygous. One dominant from one parent, one recessive from the other parent. We could have 16 traits, but if parent A was homozygous and dominant for all 16 traits, and parent B is homozygous and recessive for all 16 traits, the F1 offspring are going to be heterozygous for all 16 traits. If we cross a heterozygote with another individual with that same heterozygous genotype, they both have a big P and a little p, well now this big P could fertilize that big P and we end up with two dominant alleles. That same big P could fertilize the little p and we get a heterozygote, and that's it. This little p gamete could be fertilized with the capital P and we get another heterozygote. And then lastly, this little p could fertilize with this little p. The two recessives come together, and that is how we get an F2 individual that looks just like the parent. This white color skipped the F1, showed up in the F2. So we only have two phenotypes, two different physical appearances. They're either purple or they're white in a 3-to-1 ratio, but the genotype ratio is different. We have one homozygous dominant to two heterozygous to one homozygous recessive. So that heredity is particulate. There are individual inherited particles, and that we have to have 2 hereditary particles for each trait. We now know those are alleles, and that those alleles come apart during gamete formation so that we can pass them independently, and then it is random which 2 gametes come together to produce the offspring. And that was tested by getting each of the F1 hybrids to have one of each of both types of particle. They got a test cross going that we'll see in a moment, and then we can detect unknown heterozygotes. Because if we have a purple flower without knowing its ancestry, we don't know if that purple flower is homozygous dominant or if it's heterozygous. And there's genetic ways of determining that. If we have this F1 that has 1 purple and 1 white allele, and we cross it with a flower that we know is true breeding recessive, then we end up with a 1-to-1 ratio of things that look purple and things that look white. And that is showing how to set it up with the Punnett squares, because Dr. Punnett figured it out, that if the parent has 2 purple alleles, each gamete has half the genetic information of the parent. There's 2 P's in the parent, so a gamete can only have 1 P. This is another homozygous, but it's lowercase. It has 2 alleles for that color of flower. The gametes get 1 allele each, and they're identical, but we still put them down. And no matter which of the purple parent gametes fertilizes which of the white parent gametes, the individuals are all in the F1 generation heterozygous. And then if we have the 2 heterozygotes, the gametes from this individual is either a capital P or a smaller case p. These 2 have the same genotype for that trait, and so the same gametes are here. And now if we were to fill this in, we would have 2 big P's, or homozygous dominant, here. We'd have a big P and a little p here, a big P and a little p here. Those are our 2 heterozygous. And then we'd have a little p and a little p here. That would be our homozygous recessive. Determining if we have a dominant phenotype, but we don't know its genotype because we don't know its parentage. The way to do a test cross is to mate it with a known homozygous recessive. What we don't know, is this 2 big Y's, 2 yellow alleles, or is it 1 yellow and 1 green allele? By appearance, it looks exactly the same. There'd be no way to know by its phenotype if it's carrying 2 dominant alleles or if it is carrying 1 dominant and 1 recessive. So if we mate the unknown dominant phenotype to a known phenotype and genotype of recessive, then we look at the offspring. If all of the offspring look dominant, then we know that this had to be a homozygous dominant, because the only thing it could donate, its only allele it could donate to a gamete, is dominant. So we'd have a dominant Y and a recessive Y here, dominant and recessive here. If this were heterozygous, we would have a lowercase y, and a lowercase y and a lowercase y would have given us a green pea. Since they're all yellow, this unknown genotype had to have 2 dominant alleles, homozygous dominant. If, however, we mate this unknown genotype yellow pea with the known green pea, and we get any green peas, then we know this parent can only donate green. The other green allele had to come from this parent. And that's why it's called a test cross. We test it to see, can we determine by the offspring what the genotype of the parent was. And so we use our Punnett squares to determine the outcomes of our Mendelian crosses. This is showing the same thing we saw on the previous slide. It is called a monohybrid cross because we are hybridizing, we're mixing one characteristic at a time. This plant certainly has hundreds and hundreds of other characteristics. We're only looking at its seed color. And so we have our individuals, we look at the gametes, our parents, and our offspring always have 2 alleles for each trait. Gametes will have 1 allele for each trait. We're only looking at one trait, so gametes have 1 allele. We're only looking at one trait, so parents and offspring have 2 alleles. And here is showing our phenotypes. We started with our yellow and green, and our offspring in the F2, we have homozygous dominant, 1 of those. Heterozygous, we have 2 of those. And homozygous recessive, we have 1 of those. So our phenotypic ratio is 3 yellow to 1 green. Our genotypic ratio is 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. And again, this is only possible because these traits were dichotomous. It's either yellow or green. There's no slightly blend, there's no spotted yellow and green. It's an either or situation. The probability basics, there is an exercise for that in the Mendelian Genetics Now Extra Credit Lab where we describe probability. So we can either describe as a fraction, 1 out of 3, 2 out of 7, or we can describe it as a percentage. So a probability of 0 means it is not possible to happen, and a probability of 1 is 100%. It's gonna happen. And of course we can have ranges in between depending on what it is we're studying. So the flipped coin that we use in the Mendelian Genetics Lab, representing heads for dominant and tails for recessive, every time you flip it, you got a 50-50 chance of it being heads or tails. We can do our statistics, our probability, the overall chance of 2 or more random events happening in that exact order is the product. So we're saying this happens and the next thing happens and the next thing happens. Then we multiply that percentage or that fraction of each probability. So a die has 6 sides, so the chance of rolling a 5 is 1 in 6. The chance of rolling a 4 is 1 in 6. So if you want to say, I'm going to roll my dice, what's the odds that I will have a 5 and a 4? Is 1 out of 6 times 1 out of 6, and that's a 1 in 36 shot. Many exact combination. If there's multiple ways or tries for a chance to occur, this happened or that happens, then we add the chances. So we can certainly combine them. We are not going to have to calculate any super complicated ratios. You will perform your Punnett squares and then tell me what the chances in that Punnett square of something happening. Reading a pedigree is another feature that we're going to need for our human genetics lab. By standard, a square or rectangular symbol represents a male, and by male we are talking about an individual possessing both an X and a Y chromosome. And females are circles or ovals, and likewise a female in terms of a pedigree is talking about an individual with 2 X chromosomes. If the symbol is completely shaded in, they are affected, meaning they have the genetic condition that we are studying in this pedigree. A slash would mean the person is no longer with us. And we will see later that we can also have partial shading, halfway, which means they carry information for a trait. This is a pedigree for a genetic condition called ALKAPTONURIA. It is a metabolic disorder. So we have our first generation. These two individuals had a son, and by our key at the bottom, we see that yellow shading means they're unaffected, and blue means they are affected. They have ALKAPTONURIA. Because this affected individual has two lowercase letters, that means this characteristic is recessive, and that you have to have two recessive alleles in order to be affected by ALKAPTONURIA. These two individuals are the parents. This is their son. The son has offspring with this female who is heterozygous. And so we could do a Punnett square, and we could see the probability for their offspring would be a big A and a little a, and that individual would be unaffected. The big A could go with the other little a, still unaffected. But if this allele comes together with either of these, those offspring will be affected. So there's a 50-50 chance. And we see from their offspring, well, they have a female child that is unaffected but has the recessive allele. They have a daughter who has ALKAPTONURIA. And they have a son whose genotype we don't know. But you can figure out this individual's genotype. It has a question mark because without children, it's a little harder to see. But if we follow back to this individual's parents, this uppercase A, normal allele, had to come from his mother because his father does not have a normal allele for this metabolic trait. And so there cannot be a second uppercase A. This second allele has to be a lowercase a because it has to come from his father. Because the only place this uppercase A could have come from is the mother. This daughter has ALKAPTONURIA. And this is one of those situations where we said we can do the reciprocal cross. The father passed ALKAPTONURIA to the son, but here the mother and father passed ALKAPTONURIA to the daughter. It doesn't matter which parent donates the allele. It could come from either the mother or the father. This individual is not affected, but their daughter has ALKAPTONURIA. This mother could only give a lowercase ALKAPTONURIA A, and in order for this other A, they can't both come from the mother. Each parent only gives one copy of each trait, so this individual who is unaffected has a lowercase a and an uppercase A, is heterozygous. And likewise, we can do the same here. This individual, this female, got a lowercase a from each parent and has ALKAPTONURIA. This individual is unaffected, but this one's going to be tougher. Either parent could have given her the uppercase A, and either parent could also give a lowercase a. So without seeing her offspring, we do not know and cannot determine if this female is homozygous dominant or if she is heterozygous. So far, we have looked at one trait at a time. And now, we're going to look at two at a time. We still look at each trait individually because each trait is distinct and separate from the other trait. So the genetic information for, in humans, say, the color of my eyes and my ability to roll my tongue, totally separate. Right? The ability, which way I put my right thumb or left thumb on top versus whether or not I have a widow's peak, I don't, totally separate. But we can follow them at the same time. So we still start with true breeding. All of the characteristics we're looking at are dominant in one parent, and all of the characteristics we're looking at are recessive in the other parent. We're looking at seed shape, where it can be round or wrinkled. We're looking at seed color, it can be yellow or green. Once again, we'll pick one letter to represent seed shape. We will use uppercase for dominant and lowercase for recessive. We'll pick a different letter for seed color, uppercase for dominant, lowercase for recessive. So since yellow is dominant, I chose a Y, uppercase for yellow, and a lowercase y for green. Round seeds are dominant, so they chose a capital R for round and a lowercase r for wrinkled. All dominant alleles for the yellow round seed, all recessive alleles for the green wrinkled seed. And as I mentioned before, if you cross true breeding dominant, there's only dominant alleles for every trait. With true breeding recessive, there are only recessive alleles for each trait. The only genetic possibility for their offspring is to be heterozygous, one dominant and one recessive for each trait. And so the offspring got a big Y from this parent and a little y from that parent, and a big R from this parent and a little r from that parent. And they're going to look exactly like their yellow round parent, but they are carrying recessive alleles. To get the gametes for this seed, we have to do what we call an algebra FOIL. First, outer, inner, last. So the first letter of each set, the outer letter of each set, the inner letter of each set, the last letter of each set. Meaning we take the first letter of each combo, the first Y and the first R. They're both uppercase. Here it is. Outer, the first Y and the second R. That's our uppercase Y and our lowercase r. Inner, the second Y and the first R. Lowercase y, uppercase R. Last, the second Y and the second R. And there we go. We're crossing two F1 individuals, so the gametes from one heterozygous parent will be the exact same for the other heterozygous parent. And then we fertilize by combining the letters from each gamete, and we keep the Y's together, and then we keep the R's together. By writing the letters down, we could say big Y, big R, big Y, big R. The alleles are present. They're on chromosomes. They would be swimming around in the cell, but visually, so that we can keep track of seed color alleles and seed shape alleles, we keep the same letter together. So these two gametes fertilized, there is their letter combination. If this gamete and that one, then we have a big Y and a little y, but we have two big R's. And we look at that combination all the way across our 4x4 square, and what we end up with for the phenotype ratio is 9 to 3 to 3 to 1. 9 of them will look dominant for color and dominant for shape. 3 of them will look recessive for color but dominant for shape. 3 of them will look dominant for color but recessive for shape, and 1 of them will be recessive for both. And that is the exercise with the corn babies in lab 9. We call it genetic corn. It is corn, that has been bred so that we have two heterozygous parents coming together. And in corn, purple seeds are dominant and yellow are recessive, and round seeds are dominant and wrinkled are recessive. Slightly different colors, but the way the genetic problem works is exactly the same. So with your corn babies, you would see 9 that are purple and smooth, and 3 that are yellow and smooth, and 3 that are purple and wrinkled, and 1 that are yellow and wrinkled. So this is just showing that happens because the segregation and recombination of the alleles are independent. Like, we don't have to have all the dominant alleles in one gamete and all the recessive alleles in another, because the gametes that are formed are, well, one of them is completely dominant and one of them is completely recessive, but one is recessive for color and dominant for shape. Another is dominant for color and recessive for shape, because they are independent characteristics. This is just another dihybrid cross using different traits. And this, for our pea plants, tall plants are dominant and dwarf or very short plants are recessive. And inflated puffy pea pods are dominant and constricted wrinkly pea pods are recessive. So the letters chosen were T for the dominant tall, uppercase, lower T for dwarf, right? The same letter. Since inflated pods are dominant, uppercase I for inflated, lowercase I for constricted. And we get the same kind of gametes, a dominant, at the foil, the first big T and the first I, uppercase I, outer, the big T and the lowercase I, inner, the lowercase t and the dominant I, and last, the lowercase t and the lowercase I. Crossing 2 heterozygotes, the gamete of one is the same as the gamete for the other, and we will still get that 9 to 3 to 3 to 1 phenotype ratio. The genotypic ratio is a lot more complicated. That's our standard Mendelian genetics, and it's also called complete dominance because the presence of even one dominant allele completely dominates over the phenotype. Incomplete dominance also happens. The classic example that shows up in every textbook is snapdragon flowers. You can still have true breeding red and true breeding white, and red is dominant, so you'd have a homozygous dominant red flower and a homozygous recessive white flower. With complete dominance, we would expect red flowers. It's not what we get with snapdragons. All of the F1s that are heterozygotes are pink. This is why it was originally thought that the particles blended, mixed up, and couldn't re-separate, because there were cases where the offspring looked like an exact blending. But if you cross 2 of those pink flowers, you don't get that 3 to 1 red to white. You get 1 red, 2 pink, and 1 white. Red is homozygous dominant, pink is heterozygous, and white is homozygous recessive. So the genotypic ratio is the same as the phenotypic ratio. In people, the one example I know off the top of my head for incomplete dominance is LDL receptors for cholesterol. If you have 2 of the receptors, you're homozygous dominant, great at clearing out the LDL. If you're heterozygous, you have 1 normal and 1 damaged receptor, you kind of do okay at clearing it out. And if you're homozygous recessive for that receptor, you cannot clear out low density lipolipids, the bad cholesterol. It's the same type of inheritance. So far, we have looked at things, even with incomplete dominance, that it's still this or that in terms of the alleles. There's only 2 alleles possible. There are definitely cases where there are multiple alleles. The example here is with fur colors in bunnies. There's the coat color gene, so C is for coat color. But there's 4 variants, so we can't just have upper and lower case. We can have the dominant wild type, which is for brown fur. But then we have these lowercase. There are 3 lowercase c's, and they either lack a superscript or they have a couple. So we can have a plain lowercase c, and if you have 2 lowercase c's, you end up with an albino rabbit. No pigment. Then we have a lowercase c with a superscript ch that stands for chinchilla because it has a similar coat color to a chinchilla. And you only get that if you have homozygous recessive chinchilla alleles. We can also have a Himalayan, which is homozygous recessive Himalayan variant of coat color, where it is white fur with black tips on it. But the uppercase C is dominant to the CH. It would also be dominant to the C superscript H. It would also be dominant to the regular C. But the chinchilla is dominant to the Himalayan, and the Himalayan is dominant to the albino. So lots of different genetic combinations, multiple alleles. A slightly simpler multiple allele example that you're more familiar with is human blood types. We have the A allele, we have the B allele, and we have the O allele, 3 alleles. And that leads to our blood types of A, B, AB, and O. And that is an example of co-dominance. The A allele and the B allele are equally dominant. So if you have type AB blood, your red blood cells exhibit all of the characteristics of type A and all of the characteristics of type B. They are equally expressed. I have type A blood. Now, I don't know if I have 2 A alleles, one from mom and one from dad, or I could have 1 A allele and 1 O allele. Either one of those genotypes would give me a type A phenotype. Type B blood is the same way. You could have 2 B alleles, or you could have 1 B and 1 O allele. Type O blood can only be 2 O alleles, and that's why it is the rarest blood type. You have to have 2 recessives to get a type O blood. We can have mutants, and fruit flies are here because we know a lot about how human genetics work by following genetic traits in fruit flies. They are animals. They have X and Y chromosomes to determine sex, and they develop very, very quickly. So we can follow many generations in very short time. These poor little mutants, this is a normal fly. And the mutant one, these are not antennae. He has legs on his head. Well, that's not good. But this was looking at development. We can also have drug resistance as an example of multiple alleles. Mosquitoes deliver the malarial parasite. The malarial parasite is eukaryotic, so it has pairs of linear chromosomes just like we do. And it can mutate relatively quickly, and that is why it can become resistant to drugs. It's a real problem to develop anti-malarial drugs because they keep mutating. We have not yet seen sex-linked traits. We saw an example of a pedigree where we followed which individuals were male and which were female, but it didn't seem to matter. Males and females were equally affected. And that's because the character for ALKAPTONURIA is on a non-sex chromosome. In fruit flies, the gene for eye color is on the X chromosome. And just like humans, a fly with 2 X chromosomes is chromosomally female, and a fly with one X and one Y is chromosomally male. And by tracking males versus females and then mating the different eye colors, one time the red is the male, red eye is the male, and the next time the red eye is the female, that is how they tracked that this trait was on X chromosomes. The red eye color is the dominant color, and it's the wild type. All of the others are mutations. So here we have a red-eyed male fruit fly and a white-eyed female. So the male has an X and a Y chromosome. The female has two X's. The W's stand for, is it a wild type eye color allele or is it a mutant one? And uppercase W is for wild and lowercase w is for mutant. Because again, we choose one letter to represent the characteristic, and since we're talking wild type, they chose W. The wild type eye color allele is on the X chromosome. So a red-eyed male, the wild type allele is on his X chromosome. In order to have white eyes, one has to have two mutant alleles, because the presence of wild type would give us red eyes. So she has two white alleles. So the female could only produce X's, each of which carry the white or mutant allele, and the male has an X with the wild type allele or a Y. The Y does not carry information for eye color. Only the X chromosome does. So if this sperm fertilizes that egg, we have an X with the wild type from the father and an X with the mutant from the mother. The same for this, an X with the wild type from the father, an X with the mutant from the mother. These two would be female flies because they have two X chromosomes. They have red eyes because they have one wild type eye color alleles. The males, however, cannot get an X from the father. They only get X's from their mother. So the X's have the mutant allele. The father donates the Y chromosome, and so the males have white eyes. So even though the father had red eyes, his sons have white eyes. The mother had white eyes, her daughters have red eyes. In humans, there are definitely many X-linked traits, and they can be inherited by either females or males, but the males will be more easily affected because they have only one X chromosome. So if they have the wild type dominant X, they will have the normal phenotype. But if they have the mutant or recessive X, they do not have another X chromosome to compensate for that. So here is where we would see on a pedigree, we have the unaffected, shaded in orange, the affected faded in orange, the unaffected in blue, and now we have a carrier. And so it's a 50-50, half blue, half orange. This father has a dominant or a wild type X, so he does not have the trait. The mother is 50-50, meaning one of her X's is wild type, but one of her X's is the mutant, the recessive allele. She does not have the illness because she has one good dominant X. But she is called a carrier because she has an X chromosome that could be passed to her offspring. And so depending on which of the sex chromosomes come together, along with all the other chromosomes, we could have an unaffected son because he gets this X chromosome from his mother. And I know he got his X chromosome from his mother because his Y chromosome had to come from his father. We could have an unaffected daughter because each parent gives her an unaffected X. We have an affected son because the Y chromosome comes from his father and he got the affected recessive allele from his mother. Or we could have an unaffected daughter, but she is also a carrier because each parent gave her, or father gave her the dominant X, and the mother gave her the recessive X. We can have lethal alleles. I have a cute picture of a Manx kitty. He's got a lot of stuff going on. He's got heterochromia, 2 different eye colors, but that is not the lethal allele. The Manx cats don't have tails. They're just born that way. And all Manx cats are heterozygous for this M lethal. If you have 2 homozygous dominant M alleles, normal house cats with tails, 1 dominant, 1 recessive / lethal allele, they're healthy cats, but they don't have a tail. But if you get 2 of these lethal alleles, those cats die. They're not eliminated in utero, but they do dramatically shorten the lifespan, and so it's considered a lethal allele. So if you cross 2 Manx cats, heterozygotes, you're going to get 2 Manx to 1 normal because the other one dies. I'm going to mention briefly a trihybrid cross, only to show why we don't do them. A monohybrid cross, we're only following 1 trait, but we end up with a 2x2 square because each parent gives 2 different gametes. A dihybrid cross, we're following 2 traits, but each trait has 2 alleles, so we have a 4x4 grid. And if we're doing a trihybrid cross, we would have 3 traits, but each allele has each trait has 2 alleles, so we have 2 to the 3rd power, 2 alleles raised to the power of how many traits, and that's an 8x8 square. I don't want to draw an 8x8 square. I don't want to fill in an 8x8 square. You don't want to watch me do that. You don't want to do it. That's why we don't do it. Because the how many sides you're going to get, your number of gamete possibilities, is 2 raised to the power of the number of traits you're looking at. So far, we've talked about possibilities, and that's based only on the number of alleles. And then we see what the individuals look like. But we know from meiosis that crossover happens. And so we can get individuals that have new combinations of alleles on their chromatids. And that will also contribute to genetic variety. So we know the way that those homologous pairs hang out at the metaphase plate during meiosis 1, whether it's all the paternal on one side on the left and all of the maternal on the right, or all the maternal on the right and all the paternal on the left. Each pair can switch which side the blue and the red are on. That is variety. But we also get genetic variety from crossover. And there's really no way to calculate how many combinations there will be because crossover is also a random event. Epistasis is another factor that can affect how the alleles result in their phenotypes. How does the genotype get to a phenotype? Epistasis. Stasis means to be still. No effect. Epistasis is we're acting around that a little bit. So we're looking at mice, and a mottled brownish coat called agouti is dominant to any other coat color in mice. There is a completely separate locus, separate gene, that is responsible for how much pigment gets made. A dominant C allele means you're going to make pigment. Whatever your allele for color is, you will make that color. And a recessive allele says, I don't care which coat color alleles you're present, you're not going to get to have any pigment. So if you're homozygous recessive for the control pigment production genotype, you're going to be albino. No pigment will be made no matter which pigment alleles are present. So it is epistatic. It's hanging out around and it causes stasis. It stops the production of pigment. So if we have two mice that are agouti, their coat colors are the dominant color, and they are heterozygous for the control locus as well. So having one dominant control means they got to express their dominant coat color. We would see their gametes just like we did for the corn and for the peas. We would have a dominant A allele with a dominant C. We could have a lowercase recessive coat color with the dominant control locus. We could have the dominant coat color with the recessive locus. We could have both recessive. And since these mice have the same genotype for these two traits, they have the same gametes. The combinations of the letters would be the same, but we don't get 9 to 3 to 3 to 1. We get 9 to 3 to 4. What? The 3 and the 1, the completely recessive, get all rolled in to this albino because of the two lowercase control locus. Homozygous recessive for the C means it doesn't matter what your coat color genes were, you're going to be albino because the lowercase c controls or prevents the production of the pigment.